Mr. Sandman, bring me a dream. Make him the cutest.
Thank you very much for coming. Last lecture of the lecture series tonight, so a little bit sad, but there you go. We'll have another superb series of lectures during first of all year next year. First of all, there are dishes on the foyer for if anyone would like to donate to our charity, that is Maggie's. We've got apologies from Professor David Walker, Mrs. Sharon Scriven, Doctors David Bogard and Julie Rutter. The minutes of the last meeting are available in printed form outside the foyer. At long last, I've managed to create a podcast from Sir Harry Burns' lecture last month. The tech wasn't working quite, the tech wasn't working at all, but fortunately I had a video camera right at the back of the lecture theatre, so the sound is a little fainter than normal, but it's quite acceptable, I think. So that's available to see on iTunes. The next meeting, or the final meeting of the year is, of course, the AGM on the 10th of June, starting at 7 o'clock. On to tonight's meeting. It gives me very great pleasure to welcome Professor Russel Foster, who's Professor of Circadian Neuroscience at Oxford University. I'm also delighted that his daughter Victoria has joined us tonight. She is studying zoology at Nottingham University. Professor Foster's research interest spans visual and circadian neuroscience. I'm going to tell you more about it because I'm sure that's exactly what he does. Professor Foster was elected a Fellow of the Royal Society in 2008, which is a fantastic achievement. But having said that, I think it would be difficult to top yesterday when Professor Foster was at Windsor Castle and Prince William gave him a CBE. And the only thing that really that could top that, I think, is an invitation from Mackay to So it gives me very great pleasure to introduce Professor Foster and his talk, Regulating the Body Clock and the Unexpected Role of the Eye.
Well, thank you very much indeed, Geoffrey. It really is a genuine honour and delight to be here. I know when Geoffrey wrote to me some time ago, I was very much looking forward to this, not least because, indeed, I've come to know Nottingham well, because a certain family member is here studying and absolutely loves the city and is enjoying a degree in medicine. So, of course, the title for this evening is Regulating the Body Clock, the Unrecognised Role of the Eye. Well, actually, I've changed the title to Significated Relevance, Basic Science and Clinical Application. I will go over the eye in some detail, but I thought it might be fun for this audience to just sort of step back, rather than drill down into some of the cellular, molecular mechanisms that we've been discovering over the past few years, but to place the eye into a broader sort of neuroscience context. And so what I've decided to cover is an introduction to the 24-hour circadian rhythms, these internal 24-hour rhythms that are fine-tuning so much of our physiology and behaviour. Then drill down in a bit more detail on the neuroscience of sleep as an example of one of these profound 24-hour rhythms and oscillations. Then talk about sleep disruption, what happens when these circuits and various drivers become disrupted, and some of the consequences of sleep disruption. And of course we're all aware of the inconvenience of not sort of falling asleep at the appropriate time. But as you'll see, sleep disruption now is being associated with a whole raft of broad health issues. And then talk about the eye and its special role in regulating sleep and circadian rhythms, and of course the potential impact of eye disease on all of these topics. And then finish very briefly with an emerging area, which is the fascinating association between sleep disruption and mental illness. Many of you, of course, in this audience will be familiar that so many mental illnesses are associated with severe sleep disruption. and how might that arise, and what might we do about it, and indeed learn from those observations. We're going to cover a lot of ground, so if I say something that's incoherent or needs clarification or is just downright stupid, please raise your hand, attract my attention, we can clarify, hopefully, and then go forward. So let's kick off with these 24-hour rhythms. And the fact that these 24-hour rhythms, or circadian rhythms about a day, these clocks and sleep processes generally have captured the popular imagination. As you will be more than aware, any Sunday supplement, or indeed most magazines these days, will have some reference to sleep and clocks. But I have to say, sometimes it's not always helpful. And I thought I'd just share an example of this. In the Daily Mirror, time to set your body clock. And I was quite excited that they were paying attention to my area of biology. And I was quite happy working with Beth Gibbon, and I was comfortable with natural rhythms, rule our bodies and dictate the best times for a range of activities. And here's our countdown. Decidedly less comfortable with 10 a.m. have a bikini wear, which was, an injection or a visit to the dentist, basically anything with an ouch factor. Pain intensity is at its lowest between 8 and 10%. It's not entirely clear why, but it's probably not because brain receptors aren't as alert as they are later in the day. And I promise you I never said that. And I certainly didn't say 6:30 p.m. heralds the start of 2.5 hours. So if you're feeling a little frisky at the moment, you've already had the blues, you'll have to wait for a little bit longer until you can indulge your passions. But what all this nonsense is referring to is some genuinely extraordinary biology. Every aspect of our physiology and behaviour is being adjusted, fine-tuned, to the varying demands of activity and rest in the day-night cycle by an internal clock. And I just want to sort of give you some examples of these 24-hour oscillations in physiology and behaviour. And essentially, the axes are similar in all of these, and you have a 24-hour time axis along the bottom from 2 o'clock in the afternoon to 2 o'clock in the afternoon, and from roughly 10 p.m. To 7 a.m., so this would be the sleep phase. And you see that there are really quite marked changes, as you will be aware. There's about a degree drop in core body temperature. This is not simply due to activity, but this will happen if you or I went to a dim, dark cave, constant light, constant temperature. The clock would still give this change. Now, clearly, activity will change core body temperature, but it will be superimposed upon this moving, internally gated baseline of temperature. And indeed, that drop in core body temperature is actually rather important for sleep initiation. If you block that drop in temperature, it's more difficult to get off to sleep. In anticipation of activity, core body temperature rises for increased metabolic rate and increased muscular efficiency. Another one here, urine volume, luckily, is low overnight and, of course, rises sharply in the morning. Growth hormone is fascinating. Almost all growth hormone is released during the first half of the night, and we'll touch on that a little bit later. We'll talk about blood pressure and alertness in some detail later. The hormone melatonin from the pineal is this sort of biological marker of the dark, and is a very useful marker of the hands of the clock. And you'll see an example later on in humans where we measure the peak of melatonin as a marker of where the clock actually is over the 24-hour day. Cortisol shows that beautiful sharp rise in the morning, again in anticipation of activity. And then, of course, this profound 24-hour rhythm of sleep that we'll discuss in some detail. Let's now drill in a bit further and consider systolic blood pressure, alertness, and sleep. And what you'll notice with this curve here is that there's a very sharp rise in systolic blood pressure between 6 a.m. and 12 noon. And of course that is driven by the clock, but also activity. But let me stress, if we were to lie constantly on a bed and show no activity at all, again we'd see that marked rise in core body temperature in the morning. That correlates really rather well with some beautiful work from my colleague Peter Rothwell, who's looked at the frequency of stroke. So here we have 24-hour time base, and the frequency of stroke in a large population. And you'll see between 6 a.m. and 12 noon, there's a 49% greater chance of having an ischemic stroke than any other time of the day. And this illustrates, I think, rather nicely two really important clinical aspects of this internal clock and these changes in physiology. Because knowing that those events occur with that really quite marked periodicity, we can think about the timing of medication. So if you want anti-stroke medication, anticoagulants, for example, then the best time for the body to receive those would be prior to that 6 a.m. to 12 noon window. And of course, if we know that this is when we're going to receive most of the strokes throughout the day, then the healthcare services can be prepared in advance. That's where you would allocate your resources, for example, to be ready for that window of strokes in the morning. Actually, overall, there's a 29% greater chance of dying between 6 a.m. and 12 noon of natural causes. So tomorrow, when you look at your watch and it's past 12 o'clock, you can actually satisfy yourself that you've actually survived the most dangerous part of the day. Alertness.
Can I ask a question?
Yes, of course.
Is that across all age groups? So
Yes.
teenagers, for example, spend a lot more time in bed in the morning if they can. Is their systolic blood pressure rise a bit later in the day, or is it the same?
It's an extremely good question, and I will touch a little bit on timing. But I took out the slides on teenagers. After around about the age of 10, there's this tendency to go to bed later and later and later. Peaking in males at about 21 and a half, females about 19. And then there's a turn. Males go to bed later for longer than women. And then by the age of 55, 60, we're getting up and going to bed at about the same time pre-puberty, around about 10 or 11. And those changes seem to be very broadly driven. The data aren't particularly good by those big changes in the estrogen-testosterone axis. But you're right, there is individual variation in these. But that was just all of the subjects that Peter had available. If we then look at alertness, I think we'd all intuitively know that our ability to process information, 4 to 6 o'clock in the morning might not be great. But I just wanted to illustrate how impaired we are. These are not data by us, but are reported in this very nice review by Joe Arendt. And what we've got on this axis here is a drop in cognitive performance, and it was the ability to perform a whole range of different cognitive tasks. Details are unimportant. And in the first study, essentially it was increasing alcohol, blood alcohol concentration. And no great surprise, as you increase blood alcohol concentration, cognitive performance declines. And in the UK, I think it's still 0.8085 when you're legally drunk, and that produces a decrement of around about -15. Now let's do the same cognitive performance tasks, but against clock time, against time of day. A little bit of a dip in the middle of the afternoon, and that can be quite pronounced in some individuals, and we can discuss that later on. But after around about 10 o'clock, there's a very marked drop in cognitive performance. So this low 4 to 6 o'clock window here. But the point I want to make is that your ability to process information here is worse than if you consume sufficient alcohol to make you legally drunk, which of course correlates very nicely with the highest risk of road traffic incidents, that's the greatest chance of actually having an accident. Now, in these particular experiments, these individuals were kept awake throughout the entire experiment. So you can simply say this isn't a clock, this isn't an underlying mechanism particularly, but this is simply fatigue. But it's not that, because you see these people have actually been awake longer than these people, and you see the clock-driven rise in cognitive performance. And in fact, you can get away with this. These individuals will probably bounce up more or less to reasonable performance after one day. After 36 hours to 48 hours, then you really start to see the fatigue kick in many individuals. And then we move on to sleep. This is profound behaviour, and let's put sleep into a broader context. It's that most obvious of our 24-hour rhythms. These are data from the United States from 2011, but across our entire life, 36% of our entire life will be spent asleep. And in terms of time spent, it is the single most important behaviour that we experience, and everything is completely dwarfed by comparison. So work-related, 16%, watching TV, 11%. Household work, that varies. When Victoria is at home, that goes up a lot. But on average, I guess it's about 8%. But let's again drill into that 36% and give you a sort of a tangible illustration of what that means. I don't know if anybody's been privileged to celebrate 60 years of marriage. If you have, that's a remarkable achievement, but it's worth bearing in mind that during those 60 years, 21 and a half years sleep. So I think you really only get full credit for what But the point I hope I'll convince you of later on is that the quality of that 21 and a half years of sleep will dictate to a large extent the quality of those 38 and a half years during the wake state. The other point I'd like to make early on is the modern attitudes towards sleep are in such marked contrast to the pre-industrial era. And I just sort of illustrate a few Shakespearean quotes. I mean, you can probably all think of loads yourself. And as you think back at the plays and the sonnets of Shakespeare, there are loads of allusions to sleep. And the glorious words of, enjoy the honey-heavy dew of slumber. O sleep, O gentle sleep, nature's soft nurse, how have I frightened thee. Just beautiful. And, of course, a contemporary of Shakespeare, Thomas Dekker, sleep is the golden chain that ties health and our bodies together. Extraordinarily prophetic, back in the 1600s when he said this. And we contrast the pre-industrial era with the late 19th century, early 20th century. And essentially, the invasion of the night was made possible by Thomas Edison, who commercialized it. He didn't invent the light bulb, but he commercialized it. And we had, for the first time, very cheap light at night. And it changed it made the Industrial Revolution to some extent possible, or the second phase of the Industrial Revolution possible. And of course, with the invasion of the night, sleep became the first victim. And what's also fascinating is that from this point on, our attitudes towards sleep changed profoundly. And they're perhaps best encapsulated by Edison himself, who said, sleep is a criminal waste of time, an heritage from archaic days. And we're stuck with that attitude still. So what I'd like to do next is, having set the stage, is now consider that criminal waste of time in a little bit more detail. So, of course, if we compare the sleep and the wake states, we tend to think of the complexity of the wake state and ignore the extreme complexity that we see during sleep. If we just do an EEG, of course, during the wake state, we have that low amplitude, high frequency that you see across the wake state. But during sleep, there's great variety. So during light sleep, you have this sort of basic theta pattern, little bursts of activity, K complexes, and sleep spindles. During light sleep, as one descends further into sleep, you get the slow wave activity as a result of the synchronous firing of multiple populations across the brain. And then, of course, we bounce from deep sleep into REM sleep, often called paradoxical sleep, eye movement sleep, where you have the atonia, paralysis from the neck down, but the eyes are moving in their sockets very rapidly. And this, of course, is associated with the most vivid dream sleep that we experience. Slow wave sleep has been most recently associated with memory consolidation and information processing. If we think about what's going on, then in the brain itself, we have memory consolidation and information processing. It really is worth emphasizing that the brain isn't simply laying down experiences and throwing them back, almost like sort of a recording. But if you want to come up with innovative solutions to complex problems, a night of sleep has been shown to enormously enhance your capability. I won't bore you with the experiments, but they are really profound. And sleeping on it, which is what our grandparents told us to do, actually really does hugely improve information processing. The brain is also regulating a range of other sorts of processes. So, for example, toxin clearance. I don't know if some of you saw that extraordinary paper about a year, 2 years ago, showing that actually during sleep, beta amyloid is packaged up and is prepared to be got rid of during the sleeping state. And of course, it's build-up of beta amyloid that's been associated with dementia and Alzheimer's. And I think they push the data too hard by saying if you don't get enough sleep, you have high levels of beta amyloid and therefore you're more susceptible to dementia. That was the point they're trying to push. We don't know that. But it was a reasonable extension from their findings of what's going on in the brain in terms of toxin clearance. Growth hormone, of course, is associated with tissue repair, and of course it's that big spike that you see during the first part of the night, not of course in the brain, but throughout the rest of the body, and of course it's being regulated by the brain. The rebuilding of metabolic pathways, a whole slew of genes only associated with the metabolic axis are turned on during sleep, and also energy replenishment, the building of energy pathways that have been diminished during the active state. So we've seen a great deal of time is spent asleep. There's lots of different sleep states. It is not simply the brain turning off. In fact, some areas of the brain were active during the sleep state than the wake state. And we see that during sleep, a whole raft of different things are going on. And this is not all of them by any means. No great surprise then that sleep arises as a result of an interaction between all of the key, in fact every neurotransmitter within the brain at some level is involved in sleep. The histamines, of course, those drowsy antihistamines that we'll again touch on later on, that people use to self-medicate. That's the pathway that you're hitting there. And, of course, multiple brain structures. And again, just to illustrate that point further, we peel away the cortex and we look at the mid and the hindbrain, critically involved in sort of the ascending arousal pathway, and that's turned on and off by critical structures within the hypothalamus, and of course the hippocampus and thalamus very much involved, and of course the cortex. So the point is that sleep is a global brain event. And let's put those various statements into some sort of framework. Because I think the way that sleep has been taught is a whole bunch of pathways and stuff going on, and it's not particularly coherently put together. So I just wanted to sort of frame what's going on in the brain during the states of activity and consciousness and sleep. And so during the wake state, the hindbrain, the thalamic pathways, are bathing the cortex with all of the monoaminergic and the cholinergic neurotransmitters and driving the wake state. That is largely, but not exclusively, of course, regulated by a relatively tiny structure within the anterior hypothalamus, the lateral hypothalamus, which is releasing orexin, hypocretin, and of course it's defects in orexin that give rise to narcolepsy. And the defect in that pathway means that this is very variably turned on and off, and so you can go into that catastrophic sleep state because you've lost that incredibly important drive from the lateral hypothalamus. Really a tiny cluster of neurons within the brain. During the wake state, monoaminergic pathways then feed back and turn off another tiny little structure in the hypothalamus, the ventrolateral preoptic nuclei. And this is called the sleep switch, and you'll see why very shortly. In fact, now. Because the sleep drivers, when they're turned on, and we'll talk about those in a bit more detail in a moment, activate the velpo. The velpo then inhibits this entire lateral hypothalamic drive. So you get that entirely turned off. And adenosine within the brain has been associated as a key element in that sleep driver. So adenosine, the breakdown product of ATP, so it's a good sort of marker of energy expenditure. And of course caffeine is interfering with adenosine receptors, which is why you genuinely feel more alert after you've taken a cup of coffee, because you reduce the drive of adenosine on the ventrolateral preoptic nuclei. In addition to turning off the lateral hypothalamus, the velpo also projects to all of those monoaminergic and cholinergic systems within the brain. It's extraordinary, tiny little structure is going all over the place. And via GABA and ghrelin, actually turning off the release of those neurotransmitters. There are two sleep states, which I haven't gone into any detail. But the point is that structures within the mid and the hindbrain sort of produce a 60 to 90 minute flip-flop between non-REM and REM. That's really rather interesting because during REM sleep, you'll note that whilst during non-REM, all of the neurotransmitters are turned off to some large extent, but during REM sleep, acetylcholine is actually turned on. And so, in fact, it is at levels in REM sleep which are equivalent to the waking state. And in addition, you've got that paralysis, that atonia from the neck down. And of course, REM behavioural disorder is a disruption of that pathway where individuals will act out their dreams, for example, in REM sleep. There was a famous case recently where an Australian man killed his wife and claimed to have a REM behavioural disorder, and therefore he was tested and he did, and he got off, completely scot-free. Anyway, I suppose we can discuss the debate that some later. So the point is that there's loads of different neurotransmitters and loads of different circuits that give rise to the sleep states and the wake states. But in addition to all of this lot, let's consider those sleep drivers in a little bit more detail. And our species, almost uniquely, is going to be driven by social timing. The young people here, I'm sure, were all woken up by an alarm clock this morning. Not, yes, everybody. That's why I suspect that the more senior individuals in the audience didn't need an alarm clock, because, as we discussed, this question up here, you tend to be a later type in your youth, and then you become more of a morning type as you age. So the alarm clock and social timing can force a sleep-wake pattern on that rest activity cycle. And we talked about sleep pressure. I talked about it in the context of adenosine. It's the intuitive part of our sleep. The greater we feel, the longer we've been awake, the greater the build-up of the sleep pressure, the greater the need for sleep. And that's been associated with the build-up of adenosine. If you pop adenosine into a rat brain, particularly the prefrontal cortex, you'll actually put it to sleep pretty effectively. And of course, if you block those adenosine receptors, you can keep awake for much longer. So what's happening is that the sleep pressure is building up and up and up. From the moment we wake up in the morning, it goes up and up and up and up and up, and we don't fall asleep because of a critical second timing. That is the body clock. The body clock essentially timestamps everything and is saying, now is the appropriate time to be awake, and now is the appropriate time to be asleep. So although the sleep pressure is building up, it isn't allowed to have its effects because the clock is saying, no, you should still be awake. So there's a clock-driven wake, and there's a clock-driven maintenance of sleep. And so these two things interact very, very critically. Now, of course, sometimes they can become out of sync. Jet lag is a good example of that. And sometimes, if you're sleep deprived, the sleep pressure can build up to a higher level than the clock can say you should be awake. And so sometimes that's associated with a mid-afternoon sleep, a nap, which of course pushes back the sleep pressure. The problem with a nap in the middle of the afternoon is it will reduce the sleep pressure and therefore delay sleep onset at night. So you have to be quite careful about napping, because if you delay sleep onset at night, the alarm clock is driving you out of bed, then you've constricted the sleep episode. So you wake up with sleep pressure already. And of course you can get into a cycle of longer and longer naps, and these two wonderful oscillators can become uncoupled. Now, I've talked about the clock, but I haven't defined what it is. And we now know a great deal about this extraordinary bit of the brain. I flip the brain on its back here, and if I take a section through here, and we look just above the optic chiasm, here, we see a paired structure, the suprachiasmatic nuclei. The yellow blobs here are the cells, and the red represent the fibers. This is the master clock within the brain. And if this region of the brain is destroyed, then those 24-hour oscillations I talked about are completely lost. So, for example, if this bit of the brain is destroyed, then sleep becomes entirely fragmented. So the sleep pressure builds up, the adenosine builds up, you have a short sleep episode, you then push back the sleep pressure, and then you have a series. So it's entirely fragmented. You don't have the positive waking drive and sleeping drive, that coherence that the clock provides, and indeed all those other 24-hour rhythms are gone. Now, I was involved in experiments in the 1990s that demonstrated that in animal models, and more recently we've looked at individuals with anterior hypothalamic tumours, where the tumour is swept through the anterior hypothalamus. One individual in particular we were able to follow, and 24-hour rhythms were lost as the tumour advanced through this bit of the brain. Now, what's turned out to be truly remarkable, I think one of the great achievements in neuroscience over the last few years, has been the demonstration that individual SCN neurons are capable of generating a 24-hour oscillation. So, contrary to what we thought originally, in that those cells would be coupled, they are coupled, but we thought that essentially the 24-hour oscillation was an emergent property of a neural network, and that was not the case. Individual neurons in a dish show 24-hour oscillations, which means that there's about 50,000 of these neurons that are all coupled together, which all act to produce this master body clock within the brain. As I said, destroy that region of the brain, and rhythmicity is lost. And what we used to think, up until relatively recently, is that that master clock would send some sort of signal and impose 24-hour oscillations on the rest of the body. The organ systems, all of those 24-hour oscillations that we talked about were essentially driven from this master clock. We got that wrong too. Relatively recently, and some pioneering work by Uli Schubler in Zurich has shown that essentially every cell in the body is capable of generating a 24-hour oscillation. So what we've got is this master conductor of an orchestra producing a temporal beat from which the billions of individual cellular oscillators in the organ systems of the body take their reference cue and align their physiology appropriately. And that, I think, has been an extraordinary set of observations. Now, the demonstration that a single cell can generate a 24-hour oscillation means that the clock itself must be a subcellular molecular process. And this is one of our models of it. And I know I can sense that, oh my God, please, he's not going to go through that. And you'll be delighted to know I won't. But it is, again, one of those lovely, I think, achievements, because we do have a pretty good idea of how that molecular clock is generated. But I just want to go back to the basic component parts and give you some sense of how this cellular oscillator works. And we have two key sets of genes, the per-period genes and the cryptochrome genes, and there's two per genes and there's two cry genes. They have a regulatory element in their promoter, which binds two proteins, clock and BMAL, and that triggers transcription and translation. Those proteins then form a complex, move into the nucleus, and actually inhibit that transcriptional drive. And so, essentially, they turn off the drive, no more message, no more protein that is degraded, no more complex, no more inhibition, and the whole thing starts again. So it's essentially a molecular feedback loop with the dynamics, with all those other elements that generate an oscillation of 24 hours. And it's that rhythmic production and destruction of those key proteins that provides the signal that drives those oscillations in 24-hour physiology and behaviour that we talked about right at the beginning of this presentation. Now, what's really exciting is that changes in these clock genes, which addresses, I think, the general discussion about, changes in these clock genes are now being linked to a particular morning and evening times. And just to illustrate that further, this is the sort of normal pattern that you get, and very few people actually show this. So each line here represents a day, and this is that when the individual was asleep, and most people tend to go to sleep a little bit later at the weekends. Teenagers, of course, will be going much, much later. But there are individuals who are hugely delayed and hugely advanced. Evening types, or owls, or morning types, or larks. And clock gene polymorphism, subtle changes in these genes, have been associated with being an evening type and a morning type, in addition to those developmental changes. The most extraordinary and well-described example is a condition called familial advanced sleep phase syndrome. These individuals, no matter what they've been doing, are completely shattered, need to go to bed between 7 and 7:30 in the evening, and they're waking up for 4:30 in the morning. The whole clock has been advanced through time. And actually, it's as a result of a single amino acid change in the HER2 gene. So, and that affects the stability of that protein, which means that in fact it's more stable. And what happens is that whole oscillation is speeded up in time. Absolutely remarkable. So a single amino acid change in one of those proteins can be translated through to have a major effect on the complex behavior, which is sleep-wake timing. And as I say, less radical phenotypes can arise with more subtle polymorphisms in these genes. So we've said in one of the books, by their contribution to our genes, our parents are still telling us what time to get up. Okay, so where are we? I said all of those complicated circuits and neurotransmitters, social timing, adenosine and other substances driving the sleep pressure, and then this wonderful master circadian oscillator in the suprachiasmatic nuclei, and all of the peripheral circadian oscillators throughout the organ systems of the brain and the rest of the body. But this timing system would be of absolutely no use whatsoever unless the internal clock was also locked on to the external world. Classic mismatch. You've got a mismatch between internal time and local time. Local time is defined by the light-dark cycle, which is detected by the photoreceptors within the eye, which then sets that molecular oscillation. So it's the light-dark cycle that allows the internal clock to be appropriately aligned. And you get over jet lag, largely, but not exclusively, as a result of exposure to the new light-dark cycle and the result of projections from the eye to the SCO. But the eye is doing more than just setting the clock. There are projections from the eye, which we've been studying, I don't have time to go into, which go to many of the brains, parts of the brain associated with keeping you awake and having an alerting effect. So the reason why many of us look around a lecture theatre and see people nodding off and going to sleep is not because of the content of our lectures. It's the fact that we sit our students in dim, dark environments, and they're already tired because, of course, they've had very little sleep because they're late nights. And so if we were to introduce more light and increase the levels of light, we'd increase levels of alertness. It's actually quite interesting because, of course, it's levels of alertness that can be used to delay sleep onset at night. And so we need to minimize light exposure prior to bed. And what's the last thing that most of us do? We stand in perhaps the most brightly lit room, looking into a brightly lit mirror, cleaning our teeth. And actually that will delay sleep onset. So those projections from the eye to the alerting centers of the brain are also important. But also the eye and the clock interact to regulate pineal melatonin. This pea-sized structure right in the middle of the brain produces melatonin. The eye coordinates the activity of the clock for our multiple synaptic pathway, then regulates the pineal, and melatonin is ideally only released at night. And so it's a biological marker of the dark, probably feeds back in ways we don't fully understand. In about 70% of people, it can reduce sleep latency, so it has a direct sleep inductive effect. It's very mild. But it's probably also feeding back on the clock itself. There are lots of melatonin receptors. So light comes in, hits the clock, the molecular clockwork, and then dark via melatonin probably reinforces the light signal. So they're probably interacting, but we don't really know how. Okay, the point I've tried to make is that there's immense complexity. You've got all of these various drivers and regulators. If you are a shift worker, that will be distorted. If you are drinking excessive amounts of caffeine, that will be distorted. If you are getting the wrong or inadequate light-dark exposure, the clock will not be appropriately set. And of course, if you're a teenager, then probably steroidal influences are going to affect the way that the clock aligns itself to the light-dark cycle. All of this, and then of course all the neurotransmitters and the pathways in the brain, make sleep immensely vulnerable to disruption. And of course, sleep disruption, as we'll discuss in detail in a moment, is associated with overall health problems, cognitive health problems, emotional and mental health problems that we'll discuss right at the end. And as I no need to tell this audience, all of these are fairly connected. So what I've tried to do with this sort of model is place sleep at the heart of so much of our health, and indeed sleep is being regulated by so many important factors within our environment: light, and of course social timing, and some of the agents that we experience, such as caffeine. So let's now go to sleep disruption. Are there any questions so far? Is that always... Yeah.
Does exogenous melatonin reset the body clock?
It's a really good question, and we'll touch on that a little bit when we talk about eye disease. If you have no eyes, there's no way of regulating the clock. You're going to behave yourself. And under those circumstances, melatonin has been shown, if you give melatonin 3 milligrams, 10, 11 o'clock at night, you can stabilise the sleep-wake cycle. It doesn't work for everybody, but it's the best thing we've got at the moment. If you compare light versus melatonin, the clock always defers to the light signal. And it's when light during the day and then melatonin at night are in synergy that you probably have the real efficacy of melatonin. But it's probably also having a slight sleep-inductive effect in ways we really don't understand.
So can you use it to treat jet lag?
In the studies that have been undertaken, people like Josephine Arendt would argue, and I would agree, that it speeds up the rate at which you can lock onto local time. You have to be very careful, though, because travelling west is easy. You just seek out light, and it's fairly easy to control it. Travelling east is a real problem, because the body clock, it's easy to adapt to an expanding day, not a contracting day. And you can take melatonin at the wrong time. That can actually shift the clock in completely the wrong direction. So there are rules and regulations about taking melatonin. You have to be quite careful. And people have overdosed on melatonin and thrown the clock in lots of different sorts of directions, inappropriate light signals, inappropriate dark signals, and it can then actually be worse. It can take longer to adapt if you don't get it right. There's another question, I think, somewhere. Okay. Yes, okay.
Could I just ask you about the influence of the even finer variation,
the ultradian
variation,
Yeah.
and how that fits in with
Yes. So I've sort of tended to emphasize this pathway here as sort of setting the entire sort of temporal framework. That's an oversimplification. It's very likely that under certain circumstances, some great experiments were done with rats. They're nocturnal animals. They were only given access to food during 2 hours during the middle of the day. And then their clock gene patterns were looked at. They're under light-dark cycle. The SCN, the master clock, was perfect, just as you'd expect, locked onto that light-dark cycle. However, the gut and the liver and other organ systems had uncoupled themselves from the master clock and had aligned themselves to that restricted feeding. So it's probably other signals like the glucocorticoid axis that is feeding back on that. Exercise has also been suggested to be a useful reinforcer. Exercise, particularly during the morning, has been shown to be useful in reinforcing the clock. But your question about ultradian rhythms, did you have any specific ones in mind?
I wondered whether the mechanisms were likely to be similar in terms of the,
Yes.
all of the cells within the body.
Yeah.
Whether this is a phenomenon of the brain or is it all of the body?
Yes.
And how significant it is in terms of our clinical activity.
Yeah. So for example, there are mutant mice and hamsters with a 20-hour clock rather than a 24-hour clock. Do they show similarly shortened or expanded ultradian rhythms? And the unfortunate answer is no. There doesn't seem to be a common mechanism that is driving both short and these 24-hour rhythms. There's even, and again, I wasn't planning to talk about this, but there are circadian clocks. There are clocks with a period of a year. Mammals have them. It's likely that we have a very, very damped annual oscillator. And we know most about them in the hibernating mammals. And what's truly extraordinary is you can go in and you can destroy the SCN, the master clock, and they still show annual rhythms. So there's another clock. It's not simply the 24-hour clock counting 360. There's something else which is actually producing an annual oscillation. And again, we don't know what the hell it is. So there's a temporal pattern in so much of what we do, and it's likely that the underlying mechanisms vary depending upon the period. That's a really interesting question.
Is that annual clock related to the seasons?
Yes, and we can talk. In fact, I can send you my second book on seasons of life, because it is really extraordinary. I mean, we think we're completely independent, but actually there are very statistically highly significant annual rhythms in much of our biology. That whether they're genuine clocks in humans, we don't know because we haven't done the experiments. But if you look at other mammals, as I said, like hibernating ground squirrel, sheep, absolutely, their annual reproductive cycle is both measurement of day length, but also you can actually show that there's an annual clock as well. So it's really remarkable. Okay, so let's now look at sleep disruption in a little bit more detail. And the first point to make is that there's lots of different levels of sleep disruption. So loss of attention, high levels of microsleeps, failure to process information, memory impairment, reduced cognition, creativity, can drop really quite quickly. After 24 or 48 hours, you really hit those cognitive abilities. Slightly longer term, and perhaps in terms of months and years, immune suppression perhaps associated with increased cancer risk and certainly infection, increased cardiovascular risk of diabetes 2 and metabolic syndrome. We'll talk about those in a little bit more detail later. And then in terms of brain function, again, longer term sleep disruption, mood instability, anxiety, increased stimulant and sedative use, impulsivity, and indeed a greater risk of mental illness, as you'll see right at the end of this presentation. Let's look at these three to begin with some examples here which are very chilling. This is a study from the United States showing that in the USA 100,000 crashes every year are related to sleepiness. The peak being, of course, in that sort of 2 to 6 a.m. window where people are there most losing vigilance. The Exxon Valdez, many in the audience I'm sure will remember that oil tanker that went off and hit a reef in the northwest coast of the United States. And it's very interesting, actually. Everybody assumes that the captain was drunk, and basically was drunk and hit the reef. Well, actually he was drunk, because it was asleep in his cabin. And there was a very inexperienced, very tired junior officer in charge, and he was actually being told, turn the bloody ship, and you couldn't process the information. It was so impaired. And indeed, the accident investigators determined that fatigue was a factor in the crash, and the Exxon shipping company was criticised for failing to provide a rested crew. The tragic Air India Express in 2010, which had an appalling crash landing, the accident investigators said that the pilot could be heard snoring on the cockpit voice recorder shortly before the disaster landed. It's a really good example where if you're chronically sleep deprived, you fall into a microsleep, and there's almost nothing to do about it. There's lots of examples. I'm going to horribly, God, I am overrunning already. So I'm going to give you that lovely example. And the Chernobyl accident is a classic one, where individuals in the middle of the night shift, desperately tired, disinhibited, were actually just jockeying around, saying, "Oh, I wonder what happens if we." And they had disconnected the consequences of their actions, and as a result, this appalling accident happened in 1986. This is a nice illustration of the human brain. It's an old study now, an early fMRI study, with the same individual who was fully rested, performing mathematical tasks, and you see lots of bits of the brain lighting up. And this is the same individual after, I think they were 36 to 48 hours deprived, and that's the sort of level. It's just a beautiful illustration of how impaired the brain is. And the problem is that brains like that then do this. The alarm clock drives the tired brain awake in the morning, and the second thing the tired brain will instruct its body to do is seek out caffeinated drinks. If it's a really naughty brain, it'll seek out nicotine, which of course is extremely effective as a stimulant in staying in task. The problem with things like caffeine is that they have a half-life of between 5 to 9 hours. And so if the waking day is then fueled with caffeinated drinks, then you get to late in the evening and you think, my God, I've got to get up at 7 o'clock tomorrow morning, I'm completely wired, what do I do? Well, then you take sedatives, either some sort of pharmacological sedative or alcohol, and these indeed sedate you. But they don't provide a biological mimic for sleep, biological sleep. And so, in fact, some of those really important processes, memory consolidation, the processing of information, can actually be quite inhibited by the use of sedatives. So the alarm clock wakes you up after a sedative-induced sleep, more stimulants, more sedatives. And this stimulant-sedative feedback loop is the waking and sleeping existence of many through the developed and developing world. And looking at everybody on the front row, sort of nodding, describes my life. And of course, we think of adults doing this, but actually teenagers are increasingly driving the waking day with Red Bull and Pro Plus. We talked about those drowsy antihistamines. I don't know if anybody's ever encountered Phenergan. If you ever want to waste a Saturday, take a couple on a Friday night and just see what happens. I did this. I'm amazed you can get this stuff over the counter. These are extremely effective sedatives. And of course, children stealing their parents' alcohol. And one of the most sobering talks I ever had was with a 13-year-old kid in the university at Liverpool, and she was at one of the academy schools, and sleep was appalling in that group. And I said to this 13-year-old, "What's your sleep like?" And she said, "It's great." I said, "That's fantastic. What's the trick?" Because most of my friends here are telling me they have awful sleep. And she said, "No, it's fine. I just take my mother's sleeping tablets." And I said, "Well, how do you manage the next day?" And she said, "Well, after about 2 or 3 Red Bulls, I can function." And here we have a developing plastic brain. And they're driving it using these stimulants and sedatives, and we have no idea what the long-term consequences are. Appetite. Again, I apologise for this cartoon for this particular audience, but it serves the purpose to illustrate that, of course, the gut releases the hunger hormone ghrelin, which increases appetite, promotes particularly carbohydrate consumption, opposed crudely by leptin, the satiation hormone, which is the brain, reduces appetite. In fact, this is me before and after picking up chocolate hobnobs. I wish. The problem, of course, is that sleep disruption, and Ed Van Kouter has done some beautiful studies, which I clearly don't have time to talk about, which has shown that ghrelin release is hugely promoted by sleep restriction and disruption, whereas leptin release is reduced. And so tired, sleepy people are predisposed to have an increased appetite and increased risk of obesity. And Ed Van Kouter would argue that part of the childhood obesity that we're seeing now, clearly, there's a lot more food available and all the rest of it, but since the 1950s, sleep has been hugely reduced in teenagers. And she would argue that this increased hunger drive is part of the problem associated with childhood obesity. And we can debate that later. The stress axis, of course, is extremely useful, but sleep and sleep disruption turns the stress axis on. And so you have, of course, glucose release into the circulation, reduced insulin, therefore greater vulnerability to metabolic intolerance, diabetes 2, gastric acid secretion, and so, of course, taxi drivers, classic example of having higher gastric ulcers. One thing we know about sustained levels of cortisol is that it reduces immunity. Actually, one night of no sleep can reduce the efficacy of natural killer cells by 27%. So over time, there's a very nice study on Danish night shift nurses showing high rates of colorectal cancer and breast cancer, taking all of the confounding factors into account. Could well be the reduced immunity from sustained levels of cortisol. Of course, greater levels of cardiovascular disease as a result of sustained disruption. You see that in the night shift work. Impaired memory after long-term cortisol exposure, and of course, even further anxiety, depression, mood instability. Okay, I'm conscious of the time. Graham, are we okay to go on?
Yes,
Okay, if people have to leave, then obviously feel free to leave. I want to now talk about the eye and its importance in this whole scheme that I've sort of built up. But we're going to just focus upon essentially the eye's interaction with the clock. And this goes back to the early 90s and a clear sensory biology question. The familiar function of the eye, of course, isn't as an image detector. Detecting an object either in black and white or colour against its background. But of course, the clock doesn't need image detection. It doesn't need classical vision. It needs an overall impression of the amount of light at dawn and dusk, so that it can set the internal clock to the external world. So what the clock needs is brightness detection, not image detection. And what we knew in the early 90s was that the responses to light by the visual system versus the circadian or the body clock could not be more different. So of course, in the visual system, light has to be from a particular region of space. So you have this extraordinary temporal, spatial mapping. Light I'm looking at there is being mapped very precisely to my visual cortex. The body clock doesn't care where the light comes from. It can be anywhere in a 180-degree arc. In fact, the way that the information goes from the eye to the brain, it's actually scrambled. The SCN has no idea where that information, where that light information is coming from in terms of the eye, or in terms of the region of the eye. The visual system is ultra-sensitive to dim light, and the body clock is very insensitive to dim light. Light at which we can comfortably read by it will not have an effect upon the clock. The visual system is only sensitive to short-duration light. Of course, if it weren't, our entire lives would be a blur. Whereas, of course, the clock is insensitive to short-duration stimuli, and it's only long-duration exposure that has any effect on the clock. And then finally, the time over which the visual system integrates light information, it's in the millisecond range of vision. But the body clock is extraordinary. You can get the same number of photons expanded out over 45 minutes or squeezed into 5 minutes, and it'll see it as broadly the same task. And it was these utterly different ways of processing light information that haunted me when we asked the very straightforward question, how does the eye regulate the body clock? And of course, this is a cartoon of the eye with the classical rods and cones, the inner retina, horizontal, bipolar, amacrine, and then of course the ganglion cells that give rise to the optic nerve. And our current, in the 90s, our current explanation of how the visual system grabs light to make an image simply was, for me, incompatible for an organ that was also capable of grabbing light for the regulation of the clock. So what we did was a bunch of studies on mice. And when you take a mouse with a normal retina, and here you see the rods and cones go to the SCN, and you look at the rest activity cycle. These are all cartoons, but they serve the purpose. It's a nocturnal animal, so you see that the rest activity cycle is beautifully aligned with the light dark cycle. And under these circumstances, it's in trend. Is this photoreceptor definitely the eye, or could there be something else? You cover the eyes. What you see is the clock keeps on ticking. The mouse biological clock is slightly shorter than 24 hours, so it starts its activity earlier and earlier and earlier each day. It's failed to entrain to the light-dark cycle. So what we decided to do was take mutant mice, and we see 3 different sort of mice here. All of them had massive loss of the rods and cones and had pretty crappy reduced photoreceptors that were left. All of them were blind to visual image detection tasks. What happened? So here we have that highly retinally degenerate retina. Could it still regulate the clock? And to our amazement, it could perfectly well. And okay, is there some other photoreceptor that kicks in under these circumstances? We cover the eyes and the animal free rod. So it's something in the eye. Normal circadian responses to light in a visually blind mouse. And I was young 20 years ago, and so I had the temerity to suggest, well, could there be another receptor within the eye, completely different from the rods and cones, somewhere in here? When I proposed this, the vision community responded in the following manner. Actually, it got kind of nasty, and again, we can talk about that over a glass of wine. But I will tell you one incident in the States when I was giving a seminar. Somebody stood up at the back, shouted bullshit, and just walked out. It got really nasty. And the arguments were, we've been studying the eye for 150 years. Are you seriously telling us that we've missed an entire class of photoreceptor? That was arrogance. both by them and me. But the more damaging criticism was this. Well, the circadian system can probably maintain normal photosensitivity with just a few of these degenerate receptors. This is a very simple light detecting task. You don't need much to do it. And so there were good reasons why that argument was wrong, but what we of course had to do was generate a mouse with absolutely no rods and cones. And so that's what we did by combining a rodless animal with a transgenic focus animal. What's the result? Well, I wouldn't be standing here if you didn't guess it. No rods and cones, didn't train beautifully. And when you cover up the eyes, free round perfect. So there had to be another photoreceptor in the eye. What is it? And we now know that in the absence of the rods and cones, a small number, perhaps 1 out of every 100 of the ganglion cells is directly light sensitive. It's a photosensitive retinal ganglion cell, and it's picking up that light and firing it off into the clock centers of the brain. It's maximally sensitive at 480 nanometers and uses actually a unique opsin vitamin A photopigment called melanopsin. Nothing to do with melatonin, it's very confusing. Melanopsin. And again, we can talk about that later. Here's one of those beautiful melanopsin cells, and this is a lower power image, and what it illustrates is that these cells form essentially a photosensitive net encompassing the arc of the eye, grabbing diffuse light information and firing this off into the brain to regulate the clock. So, what about humans? Are humans like mice? And actually, I asked this question at a seminar a couple of weeks ago in Canada, and the lady at the back said, I don't know about that, but my husband's a rat. A little bit more information than I wanted to know. So we have studied 2 patients in some detail, and I whizzed through the data. But essentially, as far as we could tell, there were no rods and cones in these individuals. Appalling images of the eye, no ERG, no evidence of photoreceptors using optical coherence tomography. Certainly no knowledge of light. And what you can do with such individuals is do a quick sleep questionnaire, which is very useful. And of course, that's the first screen. And then use these little devices, these wrist actigraphs, and each line here represents 2 days. So day 1, day 2. And the reason it's done like this is that you can then plot and you can see the activity beautifully aligned in this case. And you can also see sleep onset, wake, and actually take 48 hours of urine, and from that deduce a whole bunch of cycles. And here we see the melatonin peak right exactly where you'd expect it, in the middle of the night. So this is a control individual. Here's an individual who's lost both of their eyes as a result of a ghastly accident. And you see it's midnight, and 6 o'clock in the morning, and this drifting pattern. In fact, the human body clock is longer than 24 hours, so we tend to get up later and later each day. So, what about our 2 patients? And what was remarkable, this is an elderly lady, so she had some nocturnal activity. This is a young man, but there was no evidence of that drifting pattern. So, despite no visual responses in these individuals, these 2 patients were perfectly aligned to the external world. So, are humans like mice? Yes, we are. And of course, this has major clinical implications. Visual blindness need not result in the loss of all light detection by the eye, of course. And yet, in clinical ophthalmology, sleep and 24-hour abnormalities are largely and so I'm lucky enough to collaborate with colleagues in the Oxford Eye Hospital, notably Susie Downs, and we've been looking at the impact and severity of a whole range of different retinal pathologies on the ability to regulate sleep-wake timing in these individuals. Now, I won't go into the details, you'll be delighted to know as the evening proceeds, but I just will summarise the sorts of implications of this work. So there were 2 groups of patients. Patients with visual cell loss, but functional photosensitive retinal ganglion cells. And you can assess whether the PRGs are working fairly easily. If they have no rods and cones, then, and these are working, then what should you say? Well, ideally, you should be encouraging these individuals to expose their eyes to sufficient daytime light so that they can synchronize internal day with external world. That currently is not clinical practice. fact, the tendency has been, I'm persuaded by my clinical colleagues, that, well, you know, there's nothing more I can do with you. You're blind. That's it. And there has been a tendency to say, well, wear dark glasses, and often there's no good clinical reason for that, it's an aesthetic reason. And there has been an even more terrifying tendency to say, well, look, you're blind, you can't see to look after your eyes, you'll damage them, they're the source of infection. Why don't we just take them out, pop a couple of these in, and life will be so much simpler. And then, of course, what you might have done, entirely unwittingly, these individuals are already deprived of their sense of space, of course, but by taking the eye out, you've then potentially deprived them by their sense of time. And in the same way, of course, there are individuals whose rods and cones will be fine, but let's say in acute glaucoma, you may lose the inner brain. Now, the rods and cones can't talk to the brain, and neither can the photosensitive ganglion cells. They've been lost. But again, all is not lost, and I think the gentleman was asking about melatonin. Under those circumstances, time melatonin and drugs we're working on could well be the way forward for these individuals. The point is that clinical ophthalmology must appreciate the eye provides us with both our sense of space and our sense of time, and that must be incorporated into clinical practice. And it's been estimated that knowledge of these cells could improve the quality of life of perhaps 500 million people worldwide who have severe eye disease. And currently, this other receptor is being completely ignored. Okay. Do we have time for a spot of mental illness, or shall I shut up? Yeah? You all right? Okay. So, spot of mental illness. Let's now go to this axis here. And this is new stuff, and I'm really excited to share these thoughts with you. I'm keen to get your feedback. Kraepelin, father of psychiatry, way back in the 1880s, in conditions that we would now recognize as schizophrenia, described appalling sleep-wake timing in these patients. We jump ahead to the 1970s, and of course, sleep-wake timing was noted, and abnormalities were noted, but were largely dismissed as essentially the byproduct of the antipsychotics. Very interesting. 100 years previously, prior to any antipsychotics, people were talking about sleep-wake disruption, but it was with the introduction of antipsychotics then were thought to be responsible for the sleep-wake disruption. If it wasn't for the antipsychotics, then it was abnormal sleep in schizophrenia was often dismissed on the basis of a lack of work. And this is what got me into this. I was in a lift with a psychiatrist who said, my patients can't hold down a job, so no wonder they get up late, miss my clinic, and don't have friends. And I thought that was a little harsh. And so what we did was address, with my colleague Catherine Wolfe, how bad is sleep in circadian disruption in schizophrenia. Let's measure it quantitatively using those approaches that we've been looking at in humans for other reasons. And this is the, in fact, this is an unemployed individual. Fascinatingly enough, in this study that was published, we looked at unemployed, employed. No statistical difference between the employed and the unemployed, which is interesting. When we looked at schizophrenia, the results are, I think, simply jaw-dropping. This is about the best person we saw. It's midnight, and you see that much of the activity is occurring after midnight. Very, very delayed melatonin profiles. This person is apparently normally sighted, but their rhythms are drifting through time, and we go through to an individual here who shows no 24-hour patterns at all. This is the best person we saw. And I don't have time to go into the details, but these are the worst rhythms I've seen in any mutant mouse or any other human conditions. These are absolutely smashed. And again, to cut a long story short, this is shown to be independent of the medication that they were on, and independent of their social constructs. Those profound observations and our increasing understanding of the neuroscience of sleep, circadian rhythms, made us think again. And so we proposed a very simple model, which I just want to finish with, arrogantly called a new synthesis. And what we're trying to say, though, is that sleep disruption and mental illness share common and overlapping pathways within the brain. Those neural networks that give rise to normal sleep also are shared with those neural networks that give rise to normal mental health. So if you have a defect in the neurotransmitter system that gives rise and predisposes you to mental illness, you're going to have an impact upon sleep, because as we discussed right at the beginning, all of the brain neurotransmitters at some level are associated with the generation of sleep and arousal pathways. Now clearly, the sleep disruption may have an impact upon the mental health, and the mental health may have an impact upon the sleep, but the origins of this sleep disruption reside within these common and overlapping pathways. Evidence for that, we took genes that have been linked to human schizophrenia, mutated them in a mouse, and asked the very simple question, well, what's the sleep-wake phenotype of that mouse with a schizophrenia gene mutation? And the mouse showed hugely disrupted sleep-wake patterns. So beautiful evidence for a mechanistic overlap here. People have been reminding and saying, well, okay, if this is right, then what about genes that have been originally discovered as to be associated with sleep pathways? Several mental illnesses, bipolar notably, have been associated with defects in some of the clock genes. So again, more evidence for an overlapping pathway. But what about this one? Will sleep disruption precede a clinical diagnosis of mental illness? The argument is that if the mental illness is not giving rise to the sleep disruption, but there are overlapping pathways, perhaps we'll see sleep disruption prior to any clinical diagnosis of a mental illness. So I've been working with Guy Goodwin in psychiatry on kids who are either at low risk or high risk of developing bipolar disorder. These are the low risk patterns that we see, beautiful rest activity cycle, but in high risk individuals, not clinically diagnosed with bipolar, already seeing a breakdown of the beautiful rest activity profile, suggesting that indeed there is something going on here, and that sleep disruption perhaps might be an early warning of mental illness down the road. And then finally, reduction of sleep disruption might improve the level of mental illness. This axis here. This is some preliminary data by Dan Freeman, and this is the sort of data slide I'd like to finish with. And what Dan did with patients with schizophrenia was look at their level of insomnia. This is the baseline level of insomnia, and then use essentially cognitive behavioral therapy, good sleep hygiene, consolidating sleep through behavior. And he was able to halve, both post-treatment and with a follow-up, the level of insomnia. This was the level of delusional paranoia he had with that level of insomnia. And the big question, of course, is will reduced insomnia actually reduce levels of delusional paranoia? And to his great delight, he showed statistical impact on that. So, I think this is new and emerging data, but by studying mental illness in parallel with sleep disruption, some exciting stuff has emerged. We're learning things about mechanisms. Genes that were never linked to the sleep pathways, but were linked only to mental health, and have now been shown to be associated with sleep. As a biomarker, perhaps sleep disruption can be used as an early biomarker, and therefore early diagnosis, allowing potentially early intervention in mental health issues and mental illness. We don't know, but we're certainly working on those sorts of ideas. And then finally, of course, perhaps we can actually think of the sleep systems as a new therapeutic target. Consolidate sleep-wake, and you can actually clearly have an impact on the overall health of patients with mental illness, but actually maybe even improve the mental illness in itself. Okay, sorry I've overrun. What I wanted to sort of give you some flavour for is 24-hour circadian rhythms and how we think about them and their ubiquity in our physiology and behaviour. A great success story, I think, in neuroscience has been an increasing understanding of how this 36% of our biology, our sleep, is actually generated with all of those neural circuits and all those modulators within the brain. The fact that sleep disruption is so much more than the inconvenience of falling asleep at the inappropriate time, but it essentially can ripple its way through every aspect of our somatic and mental health. And then asking fundamentally simple questions. How does the eye regulate the clock? And asking and answering that question has discovered a completely new photoreceptor system within the eye, utterly unexpected. And now, that's bleeding into clinical ophthalmology. As I said, eye giving us both our sense of space, but also our sense of time. and taking that on board could have a big impact on the quality of life of very many people across the world. And then this new emerging area of mental illness, and this fascinating association between mental illness and sleep and circadian circuitry, and an understanding, studying these systems in parallel, I think will actually inform our understanding not only of mental illness but also our fundamental understanding of how sleep and circadian rhythms are generating. Thank you very much for your attention.